Welcome to this explainer. Today, we're uncovering how an entirely overlooked tiny little plant might actually hold the biological key to securing the future of global agriculture. We're talking about a story full of microscopic inefficiencies, evolutionary workarounds, and a massive breakthrough in crop science. So let's just dive right into it.
Usually it's the flashy stuff that grabs the headlines, right? You get these cinematic discoveries like a blood-stained orchid or some crazy zombie fungus parasizing trapdoor spiders. But published quietly back in a March 2026 issue of Science is something that is infinitely more consequential to our actual survival. What scientists found inside the cells of a really small, flat, totally unremarkable green plant called a hornwort, well, it could literally redefine how we feed the planet.
To wrap our heads around this breakthrough, here's our roadmap for today. First, agriculture's biggest molecular frustration. Then, evolution's overlooked hornwort bridge. 3. Unpacking the RbcS-STAR mechanism. 4. 3 paths to better yields. 5. Decoding the yield myth. And finally, number 6, the race for food security. Okay, let's start with Section 1, agriculture's biggest molecular frustration, the Rubisco inefficiency. Basically, every grain of rice, every wheat field, and every soybean crop out there completely depends on one single, stubbornly inefficient enzyme. We are talking about Rubisco. By mass, it's literally the most abundant enzyme on Earth, and its job is absolutely crucial. It grabs carbon dioxide right out of the air and converts it into the sugars that build plant tissues. But there's a massive structural flaw. See, because Rubisco evolved hundreds of millions of years ago in an ancient atmosphere that had very little oxygen, it developed this nasty habit of occasionally grabbing the wrong molecule. And since we live in a 21% oxygen environment today, Rubisco's ancient tendency to mistakenly bind oxygen instead of CO2 has become a very serious agricultural liability.
Just to put that mistake into perspective, think of it like a daily metabolic tax. When Rubisco grabs oxygen, it triggers this whole cascade of reactions called photorespiration. So instead of putting all its energy into productive growth, which ideally gets about a 75% share, this detour slaps the plant with a 25% photorespiration tax. It wastes a massive quarter of the energy a standard crop like rice or wheat captures from sunlight. It literally dismantles part of what the plant just built and releases carbon right back into the atmosphere. For decades now, this inefficiency has been one of the biggest roadblocks to improving crop yields.
Which brings us to Section 2, evolution's overlooked hornwort bridge. The thing is, we haven't been entirely in the dark here. Nature actually figured out a workaround a long time ago, but it was always just slightly out of our reach.
For decades, researchers have known that many species of algae solve this Rubisco problem by packing the enzyme into these specialized liquid-like compartments called pyrenoids. These pyrenoids basically concentrate CO2 right around the enzyme, flooding it with exactly what it needs and keeping all that pesky oxygen out. Brilliant, right? But here's the catch. There's a 500 million year evolutionary gap between algae and modern land crops. The molecular scaffolding that algae use is so alien that trying to transplant it into our crops proved nearly impossible. Enter the hornwort. Hornworts are actually the only land plants known to have independently evolved their own pyrenoids. That makes their genetic toolkit a much closer evolutionary match to our modern crops. They are essentially our biological bridge.
Let's move to Section 3, unpacking the RbcS-STAR mechanism, because this is where the biology gets really fascinating.
So when researchers from the Boyce-Thompson Institute, Cornell, and the University of Edinburgh looked inside this smooth hornwort, they expected to find this really complex suite of alien scaffolding proteins, kind of like what the algae use. But instead, they found RbcS-STAR. This is a naturally modified version of Rubisco's very own small subunit. Right at its tail end, it features this unique coiled domain called the sequestration-associated region, or STAR. It is beautifully, elegantly simple.
Here's how it actually works, step by step. First, this modified RbcS-STAR subunit naturally integrates into the complete Rubisco complex. Second, that coiled star tail acts exactly like molecular velcro. Third, because every single Rubisco molecule now has the sticky velcro tail attached to it, the enzyme molecules just start clinging to each other. They self-aggregate into these incredibly dense, pyrenoid-like condensates. The real game changer here is that the enzyme is acting as its own scaffold. We don't need to engineer a whole suite of external alien proteins to group them together.
And brilliantly, this isn't just some theory in a lab.
researchers actually grafted just that sticky star tail onto the native Rubisco of the standard laboratory model plant Arabidopsis thaliana, and no way, it worked. The native Rubisco inside this entirely foreign plant successfully clumped together inside the chloroplasts exactly like it does in the hornwort. That proved for sure that the mechanism is completely portable.
All right, Section 4, 3 paths to better yields. This proof of concept is a massive shift for a field that's been desperately racing to break that photosynthetic yield ceiling.
Right now, global science is pushing on 3 parallel tracks. Track 1 is C4 engineering, which is this deeply ambitious long-term project to completely rewire a plant's anatomy. Track 2 is directed evolution, essentially tweaking the enzyme chemistry at the molecular level so it naturally prefers CO2. And then track 3 is our hornwort bridge, or CCM engineering, focusing on structurally grouping the enzyme together using that RbcS-STAR velcro. The absolutely crucial takeaway here? These aren't competing strategies, they're complementary. You could conceptually take the hornwort structural grouping and stack it right on top of chemically faster enzymes, creating a massive compounded boost to crop yields.
But before we go declaring an end to global hunger, let's hit Section 5, decoding the yield myth, because we really need to carefully unpack some of the numbers flying around in the headlines.
You might have seen news articles throwing around this wild 60% yield ceiling figure. Now, that 60% is the theoretical absolute maximum ceiling of a fully operational system. It's not the baseline, and it's definitely not happening tomorrow. What is realistic for a near-term proof of concept over the next decade is a 10 to 20% improvement. Don't get me wrong, that is still a fundamentally massive upgrade for global food systems, but we have to understand the true scientific scale we're working with here.
To understand the gap between that 20% and 60%, Cornell researcher Laura Gunn put it perfectly. She said, "The house has been built. The CO2 pumping system that fills it with the right gas still needs to be engineered." The hornwort's RbcS-STAR discovery essentially built the house by clustering all the Rubisco together. But to get a fully functional carbon concentrating mechanism, scientists still have to engineer the microscopic inorganic pumps to actively transport and flood that house with CO2. So we've got the rooms built, but we still need to install the plumbing.
And because of all that required engineering, scientific honesty demands we take a really sobering look at the timeline. Today we're at step 1, the proof of concept. Actually adding those CO2 pumps, that's going to take the next decade. Multi-environment field trials won't realistically kick off until the late 2030s. Ultimately, widespread farmer-accessible varieties will probably arrive closer to the 2040s or 2050s. Look, basic research with a 20-year horizon is enormously valuable, but we've got to be clear-eyed about the massive distance between a lab at Cornell and an actual rice paddy.
Which brings us to our final section, the race for food security. What does all this microscopic science actually mean on a macro level for food systems in places like Bangladesh or sub-Saharan Africa?
Well, the critical number you need to know is 30 degrees Celsius. In tropical zones, where temperatures frequently push past 30 degrees, Rubisco becomes even more prone to grabbing oxygen. The photorespiration tax just skyrockets. This creates a hard physical ceiling on rice productivity in the exact regions where it's a critical dietary staple and where global population growth is the highest.
This is exactly why the hornwort bridge is such a game changer. Unlike the incredibly complex anatomical rewiring needed for C4 engineering, dropping in this RbcS-STAR mechanism is a significantly simpler genetic modification. It uses a mechanism derived from land plants, making it highly compatible with C3 crops like rice. And best of all, it can be seamlessly stacked directly on top of existing stress-resistant rice varieties. That means we can multiply the yield potential while keeping the plant's natural resistance to local challenges like high salinity or flooding.
But as we reach this incredible inflection point in crop science, we are left with a pretty provocative structural question. History has shown us that the commercial adoption of new agricultural technology is notoriously uneven. So when this photosynthesis engineered tech is finally ready in the coming decades, will the commercial adoption gap just leave behind the smallholder farmers in the global south whose very survival depends on it the most? It's a compelling reminder that cracking the biological code is really only half the battle. Getting it out into the field is the ultimate test. Thank you for joining me for this explainer on the hornwort bridge. Keep questioning, keep learning, and we'll see you next time.